It's my pleasure to introduce to you this morning Nicole Pecora. Nicole did her MD PhD at Case Western. Her PhD was with Cliff Harding and Henry Boom, working with mycobacteria. She then moved to Boston Brigham and Women's for her residency and fellowship in infectious disease and molecular microbiology. And it's there she worked with Lynn Bry, which is how I was introduced to Nicole. While she was doing her fellowship and research fellowship year, she developed both targeted bacterial and fungal sequencing at Brigham and Women's and pathogen whole genome sequencing by NGS, which was used there for clinical use in resistance analysis, plasmid tracking, and epidemiology. She was then recruited to Rochester, New York in 2016 to develop a clinical pathogen sequencing program. And interestingly enough, that's kind of how NGS was introduced at their institution, which is a little bit different path than we took. And I think that's kind of going to be an interesting story to hear, and I look forward to your talk on whole genome sequencing and clinical microbiology. Thank you.
Thanks very much for inviting me here to talk. I don't have any disclosures. So I want to start just by commenting on the amazing things that have happened in clinical microbiology since I've been a resident over the last 5 to 7 years. And by that, I'm referring to the introduction of MALDI, so protein-based strategies that have all but obviated most of our traditional biochemicals, as well as single gene and syndromic nucleic acid panels for both primary specimens and cultured specimens, which have entered in large scale. And where in all of this is sequencing? So in my mind, sequencing really falls into 3 bins in clinical microbiology. The first one, which I'm sure everyone here is familiar with already, is 16S, which is the gold standard for speciation at this point for most organisms. So by depending on how much of the gene you sequence, you can get usually down to species level IDs with this. You can use this for individual cultured organisms. You can use it to identify things out of paraffin. And if you go across a whole community of organisms, then now you get a taxonomic profile, such as in some varieties of microbiome studies. The next bin up is multi-locus sequence typing. For those of you who aren't familiar, this is sort of a 7-gene housekeeping gene barcode that will allow you to clonal type an organism. So there's, for each species, there's sort of a universally agreed upon 7-gene set. Each of those alleles will have a number, and then you're assigned a clonal code. So this gives you more resolution than genus species. It can allow for some basic epidemiology and tracking. It allows you to identify certain high-risk clones that people are aware of and look out for. But it's somewhat limited, though more and more people are going to what's called whole genome MLST or core genome MLST. And now instead of 7 genes, you're going to 1,000, 2,000 genes, which is clearly more technically challenging, but gives you a lot more resolution. Because it's newer, we don't have the same sort of universally agreed upon barcoding nomenclature that we do with basic MLST. And then the next bin is the biggest bin, and that's the whole genome. So we'll go in, most of the talk today will be focused on that and all the information you can get from going into the whole genome of an organism. And of course there, if you go across a community, now you're talking about metagenomic studies. Again, a more in-depth microbiome-based analysis. So for these two, you can get pretty far with Sanger, though the cost of NGS is coming down so low that it's probably comparable to do an NGS versus 7-gene Sanger. But for pretty much everything else, you require next-generation technology. And in today's talk, I'm really going to focus on individual complete genome and to some extent strain typing MLST. So what can microbial whole genome sequencing bring to your clinical laboratory? Well, you can get into some very, very refined pathogen identification, as I already mentioned, sort of 16S+++. You can do very interesting things with viral diagnostics because unlike bacteria, where you have 16S, or fungi, where you have ITS, D1, D2, there aren't any universal primer binding sites that will detect a whole variety of viruses. So by doing whole genome sequencing, you can do some interesting diagnostic work. And then there's a lot of interest now in pathogen discovery and occult infections, which I'm not going to go into today, but It is a topic of great interest. Lots of centers are starting to develop this in-house. There are some commercial send-outs for this, for blood and for lung infections. But that's not where I'm focusing today. I'm focusing on more sort of tried and true whole genome applications of micro, which are in the analysis of resistance and virulence factors, and in outbreak epidemiology. Excuse me. And part of what I'm going to really focus on today is that in order to really do outbreak epidemiology, you also need to think about genomic landscape epidemiology. And by that I mean knowing what kind of circulating strains, what kind of prevalent resistance factors you have in your own institution. So it's a really localized, sort of community-specific picture in order to place, if there is an outbreak, to place that in its proper context, to really know what's going on. And of course, the other more futuristic use of whole genome sequencing in the clinical lab will be microbiome-based diagnostics, which we're all excited for and hoping for, but aren't here yet. So I just thought I would mention it. So in today's talk, I'm going to share my experience from implementing whole genome sequencing in micro in now 2 institutions, both during my fellowship, where I worked with Lynn Bree at the Brigham, where we did a pilot study looking at our Carbapenem-resistant Enterobacteriaceae, and I'll show you the data from that, as well as some prospective cases and projects, both from the Brigham and now in my new position in Rochester, where a lot of the same kinds of questions came to me in both places. So there's some definite common themes about what this kind of program can bring to an institution and the kinds of questions that clinicians and infection preventionists will ask of such a program. So to get into the pilot, I'm sure you're all aware that Carbapenem-resistant Enterobacteriaceae are a major problem, classified by the CDC as an urgent public health threat, major cause of morbidity and mortality for those who are infected. There are recently some very promising drugs that have come out, but these infections are very difficult to treat and sometimes aren't treatable. So how does a cell become carbapenem resistant? Well, there are several ways, one of which is the production of a carbapenemase in the United States. The KPC gene is by far and away the most common one that we see. Though some of these others, such as OXAs and MDMs, which I'll mention today, have been fairly widespread globally, and they're starting to creep more and more and more into the United States. So a lot of our rapid diagnostics will detect those as well, even though they're still quite rare. But there are other ways you can become a carbapenem-resistant cell, such as the overproduction or dysregulation, rather, of the chromosomal cephalosporinase AMP-C in certain species, the production of certain ESBL enzymes, particularly in conjunction with a less permeable wall, or again, this production of AMP-C. And so what differentiates all these different mechanisms, or one of the ways you can differentiate these mechanisms, is by their genetic context. And what I mean by that is that some, like that AMP-C cephalosporinase, is most often embedded in the chromosome. Fluoroquinolone resistance, for example, also is a chromosomal issue. But you can have horizontally spread genes, such as those carbapenemases that I mentioned, and they can be on transposons, which can then flip between plasmids and the chromosome themselves. They can be embedded within integrons, again, going between the chromosome and plasmids, and there can be a lot of movement, also within prophages. And as you can imagine, for a particular patient, it really doesn't matter to that person if it's on a chromosome or a plasmid of their infecting strain. It's still the same treatment challenge. But from an infection prevention perspective, it's actually quite important to know this, because as you would imagine, there's a great deal of possibility for horizontal spread on plasmids and transposons. That's not quite the same if you have an element that's embedded in the chromosome. For KPCs, by far and away, their most common platform, if you will, for spread is called TN4401. It's a transposon which is quite mobilizable, comes in different isoforms, but again, it can spread between plasmids and chromosomes and back again. So how do we look for carbapenem resistance in the clinical lab? There's lots of different ways. There's obviously always phenotypic resistance testing for rapid identification of genes. The Cepheid Carba-R is commonly used. We use that. We also use the Verigene for blood isolates. And these are great tests for giving quick decisions to guide treatment and things like that. But they're not going to give you any information about the clone that's carrying the gene. They're not going to tell you about the ST type. They're not going to tell you about the plasmids or the transposons or anything else. And to do that, you need to do whole genome sequencing, which is what we did for our pilot. So we took 41 clinical isolates of Klebsiella pneumoniae and Enterobacter cloacae. We used a general clinical extractor, so this is total chromosomal and plasmid DNA. We used the NextEra library system, and all the data I'm going to show you today is all MySeq data. Most of it is 300 base pair reads, so some of our earlier runs were shorter read length. And I'll get into the analysis in a minute, but I just want to point out, as we were approaching this problem, we knew there were certain things that we would have to pull out, and these include the MLST type. We knew we'd want to look at phylogeny, resistance, virulence factors, and those plasmids and transposons. So that's where we started. And by the way, if you have any questions, please feel free to interrupt me while I speak. So this is what our Klebsiella data looks like. This side here is the phylogenetic whole genome SNP tree. And here are the MLST types. What I'm sure is popping out at you very quickly is that there's sort of a little clustery looking thing right here. And this corresponds to the ST type 258, which is the known high-risk clone for transmitting KPCs in this country. So it wasn't surprising that most of our resistant strains that carried KPCs were localized to that clone. But what you can see is if we were just looking at species and KPC, we really wouldn't be able to differentiate this group much at all. When we start looking at the transposon, we see that while most of them are on TN4401, there are different isoforms. And then when we start looking at the plasmids, we see they actually kind of fall out into a couple different groups. By kind of integrating all that information together, we were able to resolve that these two were quite close and these two were quite close by plasmids. And then this set had it actually on the chromosome, and in one case in a plasmid. And then we were really able to get a much more refined look at that population. And then we had a couple more unusual isolates that were clearly outliers. Again, it wouldn't have been obvious that these were outliers if we hadn't gone and done the whole genome. This is our Enterobacter tree. You can see here that there are kind of two clusters that are associated with the actual KPC producing organisms. And these correspond to type ST171 and ST78, which at the time we didn't know much about, but since then it's been reported by several groups that these might be a particularly high-risk hospital clones amongst the Enterobacter, so they're pretty interesting. Again, if you look at transposons and plasmids and you put it all together, this little set really remains a set. So they really seem to have all the same plasmid and the same transposon, whereas this group starts to, again, differentiate out along the lines of different plasmids. Well, actually, it also all different plasmids because the KPC is the same and the transposon is the same. So how do we use this information to support infection preventionists? What I'm showing you on the top is all Klebsiella, on the bottom is all Enterobacter, and each horizontal line here is an individual patient, and the gray circles correspond to their admissions and discharges over time. The red asterisk is when the organism was isolated. And what you can see is if you're looking at this at a particular time, a lot of these asterisks seem to kind of fall along a fairly close time window, within a close time window. And from the point of view of infection prevention, and if you were trying to figure out if you have a problem, these would all be suspicious as being part of the same cluster. If you start breaking apart that information according to plasmids and ST types and KPC variants, then actually you realize that a couple of them really are associated with each other, and that maybe this whole group isn't really a group. And that allows you to really refine your focus and look at meaningful commonalities between those strains in order to figure out what the common source might be for clusters that continue to look like clusters. The same thing down here in Enterobacter. A lot of these isolates came around almost the same month. These three really did seem extremely close even by every whole genome parameter that we looked at. Whereas these, which were close in time, were quite different. So that gave a really much more refined picture. Otherwise, you have a lot of things to control for in these patients coming in and out of the hospital and moving all around the hospital, getting procedures done and imaging and all that sort of thing. You're trying to really focus on what they have in common. And in terms of what else this kind of information can provide stewardship and infection preventionists, it comes down to the antibiogram. So the antibiogram is what the clinical microlab contributes to allow the system to make empiric treatment decisions, to have a sense of what kind of resistance rates are in the community. And we do that based on phenotypic data. I'm showing you our antibiogram for carbapenems for Klebs and Enterobacter from the time of the pilot. And if you see imipenem, meropenem is on your y-axis and ertapenem is along your x, for Klebsiella you can see 98% of our isolates were susceptible to both. And we have this small 1.5% which is resistant to all carbapenems. Enterobacter is a little bit different because we have this group that is sensitive to imipenem, meropenem, but resistant to ertapenem. When we look at what the genomic data tells us, and we have a genomic antibiogram overlaid on top of our phenotypic antibiogram, we can see that although this is technically a CRE up here, this Enterobacter, there really aren't any carbapenemases in that quadrant. So this is a different kind of class of organisms from infection preventionist standpoint, and really even from treatment standpoint, than these guys down here. All of our KPCs are true, or OXA48, our true carbapenemase producing CREs, were all pan-resistant to all the carbapenems. So that is the pilot. What we learned from this is that genomic sequencing just gave a tremendous amount of strain resolution and description of mobilization capacity of resistance. In this kind of short version of it, I'm not going into all the plasma details that we found, but we were able to really, even with short reads, get to some pretty refined plasmid descriptions. What we also found out in our discussions with infection prevention is that ruling out an outbreak is important as ruling in an outbreak, and that it also can be much easier sometimes. And that, most importantly, this was really providing actionable data to support hospital surveillance, and it was worth going forward with a prospective use. In terms of the challenges, they're probably not surprising. The analysis was the major challenge. It's not difficult to generate sequence, but it can be difficult to analyze it. And at this point, there aren't that many off-the-shelf sort of solutions that you can use for this. There are more and more now, but there's a lot of issues with microbial analysis, one of which is the fact that you often are using a different reference every time you're doing an analysis, depending on species, depending on if it's an outbreak, et cetera, et cetera. Deciding on the most appropriate SNP calling method can be challenging in order to both not undercall or overcall. And in order to be able to use this in a timeline that's useful clinically, and what we kind of decided from talking to infection preventionists is that it's really good to turn this around in a week. You really have to have a pretty modular multi-use pipeline because you can't start analysis from scratch each time there's a slightly different question. Is it a question of carbapenemase production or a clonal outbreak or something else? You need to be able to kind of put all those questions down one central pipeline and be able to provide an answer. Also, just for the validation and clinical support of your pipeline, you also need that. So, and the other major challenge is how to communicate this information to infection preventionists and ID docs and nurses on the wards who have no sense of genomics and actually often not a great sense of even the pathogens themselves. And so we also started basically a formalized reporting system, a bacterial genomics report, to kind of really highlight and distill for each time we sequenced what the distinct question is and how it was communicated. So I'll show you some examples of that. So to get into the pipeline a little bit more, I've already mentioned the kind of basic information that we want to get out. And remember, as I just pointed out, in addition to all of these things, we also have different species all the time. And having a high-quality vetted reference is not that easy to find. Some organisms that are very well characterized, there are very high-quality references. Others, you kind of have to make them yourself. So that can be quite difficult. And so currently, this has gone through many iterations from the pilot project that I showed you up to what we're using now, but the current pipeline we're using now in Rochester has basically two arms. One arm is a raw read-based SNP calling arm, which is derived from the FDA CIFSAN pipeline. And that's where we do our SNP calling from, and that's where we make our trees and our distance matrices and things like that. And then we have another arm that's all based on assembled contigs, and we use the Spades assembler for that. And then we have a bunch of modules that we can kind of call in or not, depending on the question, and that's how we stay flexible and modular. So we have modules for MLST typing, we have modules for species finding and virulence factors and things like that, and they can all kind of come in and out depending on the question. So now I'm going to go into some cases. The cases that have come our way kind of fall into two basic categories. By far and away, the most common question that comes our way is this an outbreak? Or some variety of that question. And that can either fall out by unusual species frequency, so a species that we don't normally see, suddenly we're seeing a bunch of it, and I'll show you some examples of that. Or else sometimes there's such an unusual susceptibility pattern, and that gets people worried as well, so I'll talk about that. And then the second kind of main question is more of a long-standing question that we look at with stewardship groups, and that is just the notion of surveillance and that genomic landscape that we talked about. So I'll show some data from an ESBL E.coli surveillance project that we're doing. Okay. So our first case, this case actually came our way pretty much right after our pilot was done, and a few cases of Carbapenem-resistant Acinetobacter baumanii showed up. 4 were at different locations within 3 months. 2 were on the same unit. They had similar profiles. It caught the eye of infection preventionists. They had seen our pilot, and so they asked us to look at them. In terms of the clinical questions that we're thinking about here, because we have to really be very clear about what the question is before we start throwing organisms on the sequencer, here they're fairly simple. Was this, are they clonal? Is this strain transmission? And the second question is, why are they carbapenem resistant? What genes do they have in them? And then along with the clinical questions, we have to think about how this data will be actionable. And in this case, if they're clonal, it's pretty clear what you do. You do staff education. If it gets bigger, you probably start doing environmental testing and things like that. If they're not clonal, they have different resistance genes, well, then it was just an unusual occurrence and just keep doing what you're doing. And if they're not clonal, but they have the same plasmids or genes, well, that's a more nuanced issue, and that requires a little bit more discussion. In this case, it was very straightforward, so I'm starting with the easiest case first. So within our bacterial genomics report, these are some of the elements that we give to clinicians, including the MLST table. So these are the 7 genes that I spoke about in terms of the barcoding scheme for Acinetobacter, and these are each of the 4 isolates, and they all have completely different ST types. So basically, by definition, this is not clonal. We did a distance tree, but you really don't need the distance tree to tell you it's not clonal when they have different ST types. So it was a straightforward clonal rule out. When we looked at their beta-lactamases, they were all Oxa family for the most part, which isn't surprising because that's really what's in Acinetobacter. But there weren't any Oxa 48s, which were the highly transmissible carbapenemase oxas that are great concern. And they're all different, so they're not on the same plasmid. So this was a very straightforward outbreak, plasmid, everything ruled out. This is kind of what our report looks like. This is what the format we used in Boston. I use a very similar format now in Rochester, where we just give a very synaptic report, highlighting the main findings, and then quality information and what reference strain we used and all of that stuff. We just kind of put in the bottom. Though we're clear this isn't this is an RUO.
Excuse me, Nicole.
Sure.
You didn't put that into each patient's chart.
No.
This was for infection.
No, this does not go into a chart.
Right.
Yeah. This is research use only, and we're careful about who we communicate this to, so it doesn't end up in a patient chart, because we don't. But one thing that does pop out, so it was a straightforward rule out, so it was a nice, easy first case for us after the pilot. But what we weren't thinking about at the time, though, it seems very clear in retrospective analysis, which is that when we only put 4 strains on the sequencer, we were lucky that they were very far apart from each other, or else without context, it can be very difficult really to tell if it's clonal or not, because it's not a clear dividing line between clonal, not clonal. Obviously, these were about 10,000 SNPs apart, so there was no question. But going forward, we wanted to start thinking about this notion of genetic landscape and context really with every question that we get. So now I'm showing a case from the University of Rochester where we had some Serratia marcescens in our CICU. In this case, we had 3 cases of bacteremia and closely patients that were close together over a 1-month period. It wasn't particularly resistant, so this is one of those cases where it was frequency of species that caught people's eye. And so infection prevention talked to us, and we put him on the sequencer, but this time we found every other Serratia marcescens either from that had been isolated in that unit in a bloodstream as far back as we could go, or any other unit in the hospital in the bloodstream, as well as anything else we can find basically in the freezer that we had archived, so as to give some sense of context. And this is what the tree looks like from that. So the 3 isolates in question are here, and they're very far apart. They're thousands of SNPs apart, and you can see how helpful it is to have all of these other strains there as background. So at that point, we said, well, they don't appear clonal. They weren't resistant anyway, so we weren't worried about resistance genes. And so it seemed okay. But then there were 2 more cases over the next 4 months, and this is another thing that we've learned about outbreaks, is that they're not really that as explosive as the name outbreak seems. Usually they trickle in case at a time over the course of weeks or months, and so they kind of slowly simmer. And so as they come in, we just put them back on a run, and then since we have all the data locally, which is really nice, we just put them back on the same tree. And now we had 2 more, and they popped up down here. And they're still far apart, but you can see that they are kind of occurring in the same area of the tree. And that made us start to think about possibly a high-risk clonal type. In Serratia, there is no MLST scheme, so we don't have an MLST type. But I wouldn't be surprised as we see more of these, and then if we go into public databases, hospital strains will kind of fall out in this region of the tree, and that is sort of how ST schemes originate, by noticing kind of high-risk clones, which are still 1,000 or more SNPs apart, so still not clonal, but now kind of aggregating, which is interesting. So that's another rule out. Here's another case from our again, the CICU in Rochester. Klebsiella aerogenes, formerly Enterobacteriaceae. So some of my slides might still say Enterobacteriaceae, same thing. In this case, we had 15 Carbapenem-resistant Enterobacter Klebsiella aerogenes over a 6-month period. This shows the time period. The red lines are the ward occupancy of each patient, and then the dark lines are either the surveillance culture or a clinical isolate. And the questions are, is this a clonal transmission event? And again, why are they carbapenem resistant? To make this particular occurrence more challenging, the phenotypes are really bumping all over the place. So it wasn't even completely clear from phenotype whether they were carbapenem resistant or if they were even the same. Most of the drugs were consistent across isolates, but within cefepime and within our carbapenems, some are resistant, some are susceptible, and to make matters worse, when you tested them again, they would change. And this was driving everybody crazy. So it wasn't really clear what was going on. And this was actually the first case that I had when we were up and running in Rochester. So we sequenced them, and then we found, again, everything that we had in our freezer banks from either close units or even within the hospital of a Enterobacter aerogenes, or now Klebsiella aerogenes. And we put them on the tree, and the red ones are all the outbreak strains. The one highlighted in yellow is the index patient, and they were essentially identical. So these are dotted lines indicating that these labels really are here. So they were all, compared to the index strain, within 0 to 6 SNPs off, whereas everything else we had, the closest one was 433 SNPs, and most everything else was in the thousands. There weren't any carbapenemases in them, and I'm not going to get into that here, but the fellow who worked up this case did a great job in terms of pulling out all the mutations in the chromosomal cephalosporinase AMP-C, AMP-D regulatory apparatus and porins, which explained their phenotype and also explained the reversibility of their phenotype, which is well characterized for that set of mutations. And what we also found is that although there is an ST scheme for K aerogenes, that's not very well characterized, but all of our strains were ST4 and a couple were ST93. So now we have a ruling. So yes, we told them you do have, it seems, an outbreak. These strains are likely clonal. They do not have a carbapenemase, but they have mutations in their AMP-D and porins, which are a vertical transmission problem, not a horizontal transmission problem. And then what are the actions? So they screened everyone on that ward until 1 month after their last isolate. They monitored for increased incidence of K aerogenes pretty much everywhere. They did some environmental testing, nothing really came up, and they did epidemiological tracking, and were never able to really figure out where this came from. But it seemed like it was over. In terms of the interesting ST type, we pulled out every hospital-associated strain from GenBank that we could find. which aren't that many because this isn't a very highly sequenced organism, and we put them on a tree with our hospital isolates in purple. And what we found is that our hospital isolates grouped with these other ones, kind of really indicating that this ST4 group might really be a high-risk clone of K. aerogenes that is worth keeping your eye on. And it's highly associated with some virulence islands that we also found. So there's kind of an additional story that was interesting. So that seemed to be that, but then a couple months later, it came back. And there were, I think, five isolates that came back, one of which on the tree was not related, but the other ones were on the same, essentially identical to the ones from the outbreak a few months before. So again, what's nice about this is that because, again, we had done everything in-house and we had all our comparator strains in-house, when something popped back up, we could move on it pretty quickly, sequence it, and put it back on the same tree so you can see it in the same context with the strains we had sequenced before. And it left no doubt about how closely related those strains were. Okay, so now I'm going to move into a bit of a different case. This is another one from Boston, where it's a CRE case, and there were 2 Klebsiellas and 2 CRE Enterobacters in an intensive care unit, 2 of which, 1 Klebsiella and 1 Enterobacter, were in a single patient. And then 3 days later, a Carbapenem-resistant Klebs pneumo was in another patient in a bed very close by. And then 12 days later, a Carbapenem-resistant Enterobacter cloacae was cultured from a 3rd patient, also in a bed very close by. And so the question was, are there any clonal issues here? And are there any horizontal transmission issues here in terms of plasmids and genes? So if we look at the ST table, the first 2 columns are the Klebsiellas, and they were completely different ST types from each other, so it's not a Klebsiella clonal outbreak. And what's also really interesting is because we had done our pilot study and we had sequenced every CRE Klebsiella in our institution for the 3 years prior to that, not only did we know that these were different from each other, but they weren't similar to anything else that we had sequenced there in the past. Again, that genomic landscape picture. This isn't some clone that was brewing in the hospital that was clinically, I mean, if it was brewing in the hospital, it hadn't caused a clinical infection. But the Enterobacters were a bit of a different story. So these were both ST78, so now we have to really get down to the SNP level to see if it's clonal. And from our pilot study, almost all of our KPC Enterobacters were also ST78. So it seems that there was maybe a hospital background connection between these Enterobacter isolates and what we had seen in our pilot. In terms of the Carbapenem-aces, the first 2 isolates I'm showing on this table were the Klebsiella and the Enterobacter from the 1 patient. They both had a KPC4, so they were bona fide Carbapenem-ace producers, and it seemed to be in the same transposon and in the same plasmid. So now we have 2 different species, same plasmid, one of which the Enterobacter seemed to have a clonal type similar to what we'd seen before. So the hypothesis there was that this patient had gotten the Enterobacter and maybe the plasmid had spread over to a Klebsiella. I'm not going into the plasmid types here, but it just turns out, as we looked at that plasmid, it was of an incompatibility type that's known to be promiscuous and be able to spread species to species. So that's one of the things that we look at. In terms of the Klebsiella, in terms of the Klebsiella on day 3, totally different ST type, didn't even have a Carbapenem-ace. It was just not related. It happened to be in a patient that was right next, in the next bed, but it was not related to anything. But the day 12 Enterobacter had the same KPC4 on the same plasmid. So these 3 did seem to have a connection, 2 by clone and all 3 by plasmid. So looking at our Enterobacters A and B, the question is, are they clonal with each other, and how do they relate to the Enterobacters we had seen previously in our pilot? And so we put them back on the tree with our pilot strains. And what we can see here, here's A and here's B. These are all our ST78s. They're not clonal, actually. They're not identical. In fact, some of our pilot study isolates were closer to each of these than they were to each other. But they really did have the same plasmid. So at that point, we start thinking about things like an environmental source of this clone in the hospital and sharing the same plasmid, which is a much different question than just saying that they're, patient-to-patient spread. For those of you who are familiar with bacterial plasmids and short-read sequencing, you know that they're difficult to analyze with short-read sequencing. So we collaborated with Maria Hoffman and Mark Allard at the FDA, and we did PacBio long reads on them to really make sure that they were the same plasmid. And I'm showing a MOV alignment, and yes, they were really exactly the same plasmid. So that was a really interesting case. So the third case I'm showing is now, as we left our pilot and moved forward, we continued to sequence all CREs in the hospital, just as I continue to sequence all CREs in Rochester. And when I say that something came to our eye by unusual resistance pattern, now I'm also talking about what genes they were. And over the course of our pilot, we only saw KPCs in a single OXA48. But afterwards, suddenly we had a couple E. coli's and an Enterobacter, all who had NDMs. NDMs are still fairly rare in this country, and we noticed right away that this was a new introduction, and we didn't know what the connection was. Were the E. coli's clonal? Did we get a high-risk international clone? Did it spread to the Enterobacter? Was there any relationship in terms of a plasmid that indicated that in our system, the E. coli and the Enterobacter had already shuffled a plasmid? So it was fairly alarming when we saw those. So we sequenced them all, and even though we'd never seen them before and they all came in sort of a cluster, they were all different ST types. The E. coli's were all different. Some of these patients were international, some were domestic with absolutely no healthcare international exposures. It's really very mysterious how they ended up with this organism. But they had no connection with each other. They were different clones. And then we went into the plasmids, although they had a small core surrounding the carbapenem-resistant, which has been widely reported for this gene. The plasmid context around them in terms of the other transposons and the plasmid backbone was all very distinct. So they really had nothing in common. In terms of the Enterobacter, it was actually a chromosomal integration, so it wasn't even on a plasmid. So we were able to really get to those questions very quickly, and it was just very strange luck, essentially, or bad luck, to find all of these NDMs all at once, but they had nothing to do with each other. But the good part of this is that now, if another one comes in, you can go back here and put it in this context to say, was it related to this? Did any of these manage to get out into the system and spread or not? And we'll have that background information. Okay, so my final case is to that second category of questions that are about surveillance. So I'm not sure about the situation in Minnesota, but I know that it's very common now for academic medical centers to be in a wider and wider catchment area and getting more and more specimens and samples from the region. In Rochester, our university is up here by the lake, but there's a whole series of tiny little hospitals, 10 beds, 50 beds, 100 beds, outpatient clinics that can no longer maintain the capacity for doing their own microbiology, and they send it all to us. There's a lot of logistics in that, involving couriers and compatible LISs and things like that. But one of the good parts of it is that when we think of surveillance, it's no longer within the borders of our hospital or of our close community, and we really get to look at what's going on in a wider regional look within an academic center, which is more classically what you think of as a state or public health lab. In our case, we're concentrated up here, but we get samples basically from Syracuse to Buffalo and down to the Pennsylvania border. And when I got there, we don't have a lot of carbapenem-resistant bugs in that region, but there are a good number of ESBLs, and ESBLs aren't that well characterized, and not a lot is really known about the genomic structure in our country, so I decided to have a look at that. So we have seen more and more of them over the years. I'm showing you percent of our isolates that are resistant to ceftriaxone, both amongst urines and in non-urines, and it's up 5 to 7%. And historically, although we don't have genomic background data from Rochester, historically, ST131 is a high-risk kind of international clone of E. coli that's associated with ESBLs. And specifically, it's associated with the CTX-M-15 beta-lactamase. And this CTX-M-15 is considered so much the most common ESBL that a lot of rapid diagnostics are really based just on detecting that gene. But we didn't know why we're seeing more ESBLs in Rochester. Is it just more of the same, or is something different? Is something different happening now with the introduction of a new clone that might have different virulence properties or something like that that we should be seeing? And so we did 94 isolates. Our fellow Heba Mustafa did this work, and she's preparing it now. Looking at urine and in sterile sites, and we did see a lot of ST131, which is what is showing in blue, which is what we expected to see, but we saw a very high percentage of this other clone, ST38, which really hasn't been characterized much as an ESBL uropathogenic clone. And in fact, it's a whole different kind of E. coli that's enteroaggregative and is really more associated with an intestinal E. coli. So it was pretty surprising to see. And when we looked at their whole genomes, we saw these are all the 131s here. The red ones are sterile sites, the blacks are urines. They're really, all the 131s are really, their dotted lines are just the labels. They're really very tightly associated on this inner ring of the tree. And the ST38s are out here, and they're a little bit more diverse than the 131s, because when we saw this unusual population, we thought, well, maybe there's just like a little blip or a little outbreak or something like that, but they're actually fairly diverse. When we look at the genes they're carrying, we did see a lot of CTX-M-15, which we expected to, and the red shows the proportion that's associated with ST131, which we expected to see. But we also saw a lot of CTX-M-27, which really hasn't been, which isn't rare in the US, but is more associated with Europe and Asia and things like that. And interestingly, a big chunk of it was associated with this ST38 clone. So there seemed to be a ST38 CTX-M-27 association, which was quite unexpected, and we're pretty interested in following up on it. Getting to this question of being able to look at your catchment and figuring out whether this is just kind of like an isolated little flare or outbreak, we then went and put these isolates back on our map by approximating zip code. And the red shows our isolates, the black shows the ST38s, and you can see that they're really all over the region. It's not just one isolated little area. So this was very interesting, and it made it seem that we're not looking at just some hyper-localized phenomena, that it really might be a sense of something happening on a larger scale. I already mentioned that plasmids are hard to look at with short reads. The kind of plasmid in E. coli and INCEF plasmid is particularly hard to look at with short reads because it has a lot of repetitive stuff in it. We can't do PacBio on 94 plasmids because it would just be too expensive, so we decided to trial the nanopore system and do hybrid assemblies between nanopore and Illumina on all those plasmids to see if the backbones of the CTX-M-27s and the 15s were unique, and to really get to the genetic context. And I'm not going to show all the data from that. But we had a lot of interesting results, such as almost all our CTX-M-15s weren't even on plasmids. They had all integrated into the chromosome, which was unexpected and strange. And then the CTX-M-15s, the CTX-M-27s were riding on completely different backbone classes from the ST38s and the ST131s, indicating that there's kind of two CTX-M-27 had jumped into both of those clones, is the hypothesis, and is spreading almost independently, indicating that CTX-M-27 is giving some sort of competitive advantage. So we're following up on that. So where does this all get us? So we have a significant repository of sequenced local strains, along with finished ESBL and CRE plasmid sequences. This is our genomic landscape. This is our background, so that whenever we're looking at more things, we can make sense of them. We're developing more and more of a local picture every time we work up an outbreak and we use archived or local strains, Serratias or this and that. It builds up, and your database becomes more and more useful the bigger it is. In partnership with ID and infection prevention, this really enriches the information conveyed by our antibiograms and can influence empiric therapy recommendations. I'm not going into the detail, but for example, in that E. coli project, the antibiograms for the two ST types were actually quite different in terms of standard urine UTI drugs. And so it comes together in sort of a picture of how we use genomic surveillance in the hospital. Strains come from everywhere, and they end up in your hospital. If you're in Boston, you probably have a farther flung thing than I do in Rochester, New York, but now we still have our regional surveillance. And then we have a phenotype that we start with, but once we do genomic surveillance, we can really make this very rich database, and we don't just track clones and ST types, but we can track plasmids independently and look at something like a plasmid outbreak, like I showed you with the Enterobacter and Klebsiella strains. I'm not showing you functional data today, but that's another possibility. But most importantly, you end up with a local database. You also put these things into national databases. So when I showed you that big tree of the Klebsiella aerogenes and showed you different high-risk clones, the more and more hospitals contribute that kind of data, the more informative those kinds of analyses can be. So quickly, logistics. How much does it cost? How do we do it? How many people do we need to support it? Currently, we're running about $100, $150 in supplies for each strain. Closer to 100 unless we have to repeat. So currently, this is supported by Pathology. We're trying to link it in with the infection prevention budget to make it a more routine, standard thing. The attending that covers the reports is in micro, so it's me. I have a half FTE of a bioinformatician. We're starting to look at more off-the-shelf software solutions. The fellows and the path residents are responsible for report generation for the bacterial genomics reports. That gives them experience in the analysis and the communication. We can usually turn them over in about a week because of that modular pipeline that we can put kind of everything through. And we're working to integrate this into more standard, along with paying for it, also integrating it more into standard hospital IP and stewardship programs. So this is my summary. I think I've said all these things already, so I don't need to say them again. And I just want to acknowledge a bunch of people, both in Boston and Rochester, who have helped, starting with my mentor at the Brigham, Lynn Bree, who got me started in all of this, and she continues to do it there. My fellows at University of Rochester, Heba and Adel, who did the Klebsiella and the ESBL work. Samantha Tafner is our bioinformatician in Rochester. And then several other members of both the FDA for doing our PacBio support, and the ORISE program for providing a fellow at Boston, and both of our departments. And thank you very much for your attention.
Okay, thank you. We've got time for some questions.
Hi, thank you. Great talk. In thinking about how you fund this over the long term, you talked about tying it into the prevention budget. Do you have thoughts? I guess one of the challenges facing my work is trying to project out, how many lives could be saved, how many hospital days could be averted. It's kind of an unknown problem if you're not historically sequencing outbreaks. Can you comment on that?
Actually, this is something we're trying to work on right now, because some of this Serratia and the Klebsiella were very high profile in our hospital. It had a very clear effect. Right now, people are very aware of the usefulness of this tool, and it's more likely, I think, to have traction. But in terms of actually saying, okay, how many patient days, how much, cohorting budget did you save, this and that, it's difficult, and we're trying to find some way to really put a metric on that. And once I find an answer, I'll tell you.
Yeah, please share.
Yes.
Does infection prevention have a software program that permits them to dive into all of the micro data? So they stimulate the question, they bring it to your attention, or do you have an ear to the ground where you see 2 or 3? Because they're spread out, many of the ones that you showed us over time. So who is initiating the dialogue?
It's a little bit of both. It's a little bit of both. And that's another thing that we're working on. It was very different in Boston from in Rochester because of different systems. So in Boston, we were using WhoNet, and within the microlab, we were monitoring frequencies of both species and resistance. In Rochester, they haven't had software to do that. They're starting to use the Epic Icon, and we're just the module, and we don't have experience yet. It's newer, but it's more of standard epidemiology. They tell us we have a few of these on a unit, or we just kind of see them as we monitor. Things like CREs we see right away. And we monitor bloodstream isolates. It's a little bit of both, but it's not formalized right now, and I want it to be formalized. Yeah. Yes?
I'd like to go back to your comments about the antibiograms. Can you visualize the molecular studies somewhat replacing the antibiograms to provide more personalized treatment for specific strains of bacteria?
I think it could. It's really all about scale, right? Okay. So certainly for some high-risk pathogens and high-risk species, you could. But to really do that, I don't think it'll ever replace the phenotypic antibiogram. I see it more as enriching the phenotypic antibiogram. Yeah. But you need to do regular monitoring of certain species across wards, across time, to make it kind of consistently useful as an enrichment to the antibiogram.
Do you find that your nursing homes feed in a lot of these organisms?
The nursing homes are great sources of resistant organisms, yes.
How about thank you? I got a question. I can't remember the organism, but you have 4 examples, and the second one was something where you were concerned about environmental.
Yes.
Did you pursue that, and what did you find?
So that was happening right as I left my fellowship, and I believe at that point they were talking about doing it, and at the time nothing else had popped up, so I think it was kind of wait and see. And I don't know, I didn't hear that it had popped up again, so I'm not sure. I'd have to ask. Thank you so much for your attention. Sorry, I went long. Oh, okay, this said an hour, so I thought.
That was a great talk.
Thank you so much.
Really good.
Thank you.
Do you have a